Hello and welcome to the process of mitosis explained as told by freshman students Pauline, Bucu,
Coronel,
Audra, Frogos, and Kylie.
In the life of a cell, it goes through a set of repeating events within the cell cycle. Cell division or mitosis is one phase of the cycle. If a cell is not going through cell division, it is going through interphase, the time between cell divisions.
Interphase can be divided into three stages: the G1 or gap phase, S phase, and the G2 or gap 2 phase, and is where cells spend most of the cell cycle in. G1 stands for the time gap following cell division and foregoing DNA replication. In the G1 phase, the offspring cells grow to a mature size and proceed to the S phase after doing so. This is the phase where DNA is synthesized. The next and final phase of interphase is the G2 phase, representing the time gap following DNA synthesis in the S phase on to cell division. The time the cell prepares for the next phase of the cell cycle, cell division, is the G2 phase. However, cells can exit the cell cycle, the G1 phase, and enter a state called the G0 phase, where cells do not copy DNA and do not prepare to divide. Cells that remain in the cell cycle enter the next phase of it, cell division. Cell division consists of mitosis and cytokinesis. Mitosis is the division of the nucleus that occurs during cell division. It is a continuous process that enables the uniform distribution of a cell's replicated DNA to offspring cells. This is done in four phases called prophase, metaphase, anaphase, and telophase.
As a quick recap, chromosomes duplicate and join together at the centromere during interphase.
The first stage of mitosis is prophase. In this stage, DNA is shortened and coiled tightly into rod-shaped chromosomes. The nucleus is where the hereditary material is stored, and it disassembles and reassembles each time the cell divides. Chromosomes keep DNA wrapped around proteins that allow it to fit inside cells. When already duplicated, they condense and attach to spindle fibers that pull one copy of each chromosome to opposite sides of the cell, resulting in two identical daughter nuclei. During this phase, the chromatids, the two copies of each chromosome, remain connected to one another by the centromere and the nucleus and the nuclear membrane breakdown. Centrosomes appear next to disappearing nucleus and move toward opposite poles of the cell. During their separation, spindle fibers, which are what form the protein structure that divide the genetic material to divide in a cell, radiate from the centrosomes to prepare for metaphase, called a mitotic spindle, made up of kinetochore and polar fibers.
The second phase is metaphase. In this process, the kinetochore fibers move the chromosomes to the center of the dividing cells. Once it reaches the center of the cell, the kinetochore fibers hold the chromosomes in place. The third phase is anaphase. The chromatids separate and move from the centromere towards the opposite parts of the dividing cell. Then, the chromatids become individual chromosomes after the chromatids have separated. The fourth step is telophase. The spindle fibers disassemble after the chromosomes reach the dividing cell, and the chromosomes become a less tightly wrapped chromatin state again. Then, a nuclear envelope develops around the set of chromosomes. The spindle fibers disassemble, and the chromosomes return to a chromatin state, and the nucleolus forms in the newly forming cells. The last but not the least step is cytokinesis. In animal cells, the cell membrane, which controls what goes in and out of the cell, and acts as a barrier to keep everything inside, pinching into the center of the cell. The cleavage furrow is the area which is pinched and separates into two cells. In plant cells, vesicles from the Golgi apparatus form a cell plate at the center of the dividing cell, later dividing the cell into two cells. Offspring cells in both animal and plant cells are about equal in size, where an identical copy of the cell is made. Once mitosis is complete, the end result features two identical daughter cells with the same number of chromosomes as the parent cells. In human, it would be 46 or 23 pairs of chromosomes.
Now let's review the roles of the new terms and what happens to them during the process to help break this down for you. Chromosomes. A chromosome is a strand of DNA encoded with genes, and their role in mitosis is to carry all of the genetic information. Condensed chromosomes duplicate and attach to spindle fibers on opposite sides of the cell. Centrioles. Centrioles help to organize microtubules in the cytoplasm. These centrioles move towards opposite poles of the cell with spindle fibers following. Centromeres. Centromeres have two functions in which include joining the sister chromatids and attaching the microtubules in the mitotic spindle. It attaches chromosomes to spindle fibers during mitosis. Paired centromeres split apart as daughter chromosomes are pulled towards opposite poles.
Spindle fibers. Spindle fibers form a protein structure that divides chromosomes into parent cell into daughter cell through contracting and moving chromatids to the separate cellular poles. These fibers are formed during prophase and break down during telophase. Nucleus. In the beginning of mitosis, the nucleus disappears and the nuclear membrane breaks down, allowing most of its contents into the cytoplasm. At the end of mitosis, the nuclear membrane reforms around each set of chromosomes. Cell membrane. The cell membrane controls what goes in and out of the cell. As the end of cell division nears the cell membrane closes in and pinches the cell into two separate sets of chromatids or two identical daughter cells.
How does it connect to producing and maintaining complex organisms? Well, in mitosis, more cells are produced in an organism utilized for growth and repair. It is a form of cell division in which two identical copies of a cell are produced. In single-celled organisms, mitosis is a form of asexual reproduction, where organisms produce exact copies of themselves through mitosis. While not all unicellular organisms may not reproduce through mitosis, some may go through binary fission. However, multicellular organisms rely on mitosis for growth and repair. More cells are produced during mitosis so that it continues to grow or regenerate cells to repair any damages done to that organism.
This concludes the end of our video. We hope you learned a little something new or helped you to better understand the process of cell division, also known as mitosis. Thanks for watching!